Hey guys, welcome back. We are now moving into discussing the building blocks of life, proteins. As you may have looked at kind of your pre-homework assignment in cell biology, we focus on protein structure, function, location in the cell, and how does that lead to the structure and function of organelles and cellular activity. So we're kind of starting at that level. In my molecular class, we start at the DNA, the gene level, but here we're starting at the protein level, so-called building blocks of life. I know you had some of this in basic biology, cell biology, microbiology. Okay. So proteins, this cartoon slide just wanted to hit the molecular organization of a cell. Look at our friend the cell. There's the nucleus. There looks like this is chloroplast here. That looks like a plant vacuole there. There's some Golgi there. So we'll put a little G there. Boy, I can't write with this. Various lysosomes, and I guess that's mitochondria right there. So they're showing you the different levels. So they're starting from the cell level, moving into the next level down. So we're talking top to bottom here. And the so-called level, so that was level 4, the cells, the organelles. Level 3 are the complexes, things like chromosomes. Chromosomes have your DNA, but they have specialized proteins called histones. There's a structure, a scaffolding structure as well. And there's deep organization into chromosomes, the wrapping of the DNA, is it acetylated, non-acetylated, methylated? Those are aspects of gene expression. And then we have the membrane layers. Here's the plasma membrane, and there are proteins. So it's not just lipids, it is proteins here. Here's some proteins. This looks like a channel protein that we'll talk about. Looks like there are proteins that have lipids, carbohydrates, and other attachments. And the blue here are the hydrophilic polar head groups of the lipids, and the yellow are the hydrophobic fatty acid tails. So it's said to be a bilayer. Here's layer 1, here's layer 2. And there are proteins, transmembrane proteins, okay, transmembrane, that go across. Just something handy, it takes 20 amino acids to span a typical lipid bilayer, and those amino acids will be hydrophobic. So when we do our computational, our informatic protein structure analysis that we'll get into for our inquiries, one can use that software to actually see these transmembrane domains, they're called. Any specialized subarea of a protein is said to be a protein domain here. Oh boy, it's hard to spell with this pointer. It's a domain. And these have specialized functions. So 20 amino acids here. Here's AA for amino acids. They would have to be hydrophobic for them to cross the hydrophobic fatty acid groups here. There are proteins that are on one side or the other. Those are said to be peripheral proteins. So they're stuck on either side. And when we talk about signaling, signals will actually be intercepted on these proteins on the surface. They could be transmembrane proteins, they could be peripheral. But anyway, the signals, growth factors, other kinds of signals will be received, and then that signal will be moved to the nucleus. And we will talk about cell signaling and how cell changes its function, structure, location, activities based on these signals, right? Cancer cells are receiving the wrong signals or shortly. Again, size, we're talking proteins here and DNA here about 1 nanometer. Hey, that is my French bulldog protecting me, I guess. Hey, shh, there he goes. He's protecting me. Okay, so getting back to size, we have large proteins, in about this region. Okay. So here are building blocks of life. Different proteins have different shapes. There's an association with structure and function and the shape of the protein. And these are just some examples of different proteins. Some proteins are enzymes. They usually have the ASE at the end. Okay, here's another ACE. So these glutamine synthase and adenyl kinase are enzymes. They're also structures. These immunoglobulins are known as antibodies. And I know in micro you went into detail about antibodies. It's actually 4 proteins here that makes an antibody. 4 proteins, 2 heavy, 2 light chains. So it has very sophisticated structure. Here's hemoglobin. It also is made of 2 alpha and 2 beta subunits. So these again are the biomolecules structural in the kind cell, of sophisticated structure. We can start at the lowest things layer. Like insulin and other proteins might have to be altered and changed after they're made. So for proteins, of course, it has to get to the right location in the cell. So there's a lot of things that are going on here. H and then the N-terminus shape and the structure and function. The so-called R groups. There's could be up to 20 different R groups. DNA molecule, and it's showing the lipid bilayer, plus the hydrophobic and the hydrophilic. When those are bonded together, and the hydrophobic fatty acid bonds. Okay. So if some of this is making sense, suggest you go back to that. Okay, and I know this is all from microbiology, so enzyme that does that. Kind of catch up. Again, and the enzyme keeps building. Cell biology protein, is a senior level this course, is in the ribosomes so, here. Some of this, so go back to hope your a lot of P groups in the ribosomes and catch that up. Probably bored, but and you end up getting anyway. Something I still have to try to keep everybody on the same level. So polypeptide bonds. That so here are building blocks of life. Amino acids. Notice the R groups different shapes. There's an association R1, 2, 3, structure and function, etc. And that's said to be a shape of the protein. So what we call a protein is actually a polymer. Different proteins. An amino acid is said to be a monomer. Enzymes. Okay. They usually have the ASE. For DNA, we have the nucleotides here. Okay. Here's another ACE. So these glutamine synthase and adenyl kinase are enzymes. They're also phosphate group these immunoglobulins. And if you don't know, when you add and link phosphates in micro, you kind of store energy, and when you break detail about antibodies, it's actually so building 4 proteins here that energy. An antibody releases 4 proteins, and that's said to be 2 heavy, 2 light chains. Series. So it has very sophisticated structure. Is considered a polymer. Hemoglobin. Which it also is made of nucleic acid 2 alpha and 2 beta subunits. So these again are structural. Then move to the sugars. Sugars are things like insulin and other proteins might have to be altered and changed after they're made. And And a sugar
and,
plus a sugar,
of course, it has to get to
gives
the
you
right
actually
location
disaccharide
in the cell.
They're
So
not
there's
really,
a lot of things that
a
are going on
polysaccharide
here
is more than
with
two. So
the
that makes it a little bit
shape and the
confusing.
structure and
But
function.
that's bond is called a
There's some, of
glycosidic
course, here's
bond,
the
and that occurs
DNA molecule, and
where
here's the
water
lipid
is
bilayer,
actually
the
removed
hydrophobic
and the
so
hydrophilic
the chemical equation would have
head
water
groups
being
and the
released
hydrophobic
as we
fatty acid tails.
build and store
So if some
sugars.
of this is making
That's
sense,
what
suggest you
your
go back
liver
to
and other
your notes from
organs
microbiology, basic
form,
biology, and kind of catch up. Again, this is just meant as an introduction.
and cell biology is a
things
senior
like
level
liver
course.
does
So
that.
some of this.
So we
hope
have
a lot
polar
of
head
it
groups
is
for
just
our
review,
lipids,
and
so
you're
that's
probably bored.
the
But
sugars. Now
anyway,
we're to
I
the
still
lipid.
have to try to keep
So this
everybody on the
is
same
what we
level
saw
before.
as the hydrophilic.
So here are
head
building
group,
blocks
polar
of
head
life.
group,
Different
and
proteins
then
have
these are
different
two
shapes. There's
fatty
an
acid
association
tails. So
with
there's two
structure and
carbons
function
right there
and the
that are
shape of
attached
the
to the
protein.
string of fatty acids.
And
Here it
these
is.
are just some
And
examples
a glycerol
of
has
different
the
proteins.
third
Some
carbon,
proteins are
enzymes.
and there's
They
the
usually
phosphate
have
and
the
the polar
ASE
group, and you end
at
up
the
with
end.
a phospholipid.
Okay,
Here's
a
another
bilayer
ACE.
here.
So these
in cell
booming
biology,
synthase
and adenyl
lipids have great
kinase
importance
are
as well.
enzymes. There are also
Lipids
structures.
are,
These
separate out
immunoglobulins
organelles
are
from each
known as
other,
antibodies.
you
And I
get
know
all
in
sorts of different
micro,
function and
you
structure
went into
based on
the
detail about antibodies.
lipid
It's actually
layer here, and we'll talk a
four
little
proteins
bit more about
here, different
that makes
organelles.
an antibody.
And there are
four
different
proteins,
polar
groups
too heavy, too
that
light
give
chains.
a little different flavor.
So
if you
it
want to call
has
it
very
that,
sophisticated
of the
structure.
lipid bilayer. So
Here's
hemoglobin.
the
It also
mitochondria,
is made of
two
lipid
alpha and two
bilayer,
beta
has
subunits.
some
So these
differences
again
in
are
Golgi and
the nucleus.
the
And
structural.
so that's a
kind
fine
of
point, but I
sophisticated
at least
structure.
wanted to cover that.
Things like
So the
insulin
big
and other
take-home, and I
proteins
know
might
this
have
is
to
review,
be
is
altered and
monomers to
changed after
polymers,
they're
and you
made.
certainly need to be aware.
And
Of course, it has to get to the right location
nature here
in the cell. So there's a lot of things that are going on here.
With,
The proteins are
amino
shape
acids
and the
then
structure
linked
and
by
function,
peptide bonds. There
but,
it is, and there's a
some, of course,
space-filling
here's
model.
DNA molecule, and here's the lipid bilayer,
the hydrophobic and the
amino acids
hydrophilic
hooked together.
head groups
There's
and the
various
hydrophobic
R
fatty
groups
acid
that are
tails.
going. So that's kind of review.
So if
that's
some of this
peptide
is making
bond.
sense, suggest
Here's
you
your
go back
peptide
to
bond.
your notes from microbiology.
And so
basic
here are the
biology,
naturally
and kind of
occurring
catch up.
amino
Again,
acids
this is just
divided
as an
into
introduction.
groups depending
Cell
on
biology
their
is
R
a senior
group
level
characteristics.
course. So
Some
these
of
are
this,
nonpolar
hope a
aliphatic
lot
R
of it
groups.
is just review,
Glycine,
and alanine,
bored.
proline, valine.
Anyway, I still have to try to
methionine,
keep everybody on the
isoleucine,
same
and
level
leucine.
before we. And
So
these
here
are
are building
nonpolar,
blocks of life,
and the polar
proteins
ones
have
will be
different
the
shapes. There's an
hydrophilic.
association with structure and
These are the
function
ones
and
that can
the
be found
shape of
on
the protein.
lipids.
And
Here,
these are just some
serine,
examples of different
cysteine,
proteins.
asparagine,
Some proteins
and
are
glutamine here.
enzymes.
And the R group is the purple area
at
so
the
that's
end.
the chemical
Okay,
structure.
if
You're taking
ACE,
biochemistry,
so
I
these
suspect you'll have to
booming
memorize
synthase
all
and
the
adenyl
structure, bonds,
kinase
names,
are
spellings,
enzymes.
There are also
and whatnot.
structures. These immunoglobulins are known as antibodies.
To get it close,
and I know in
micro,
here's our
went
aromatic
into,
groups. These
detail
are very,
about antibodies. It's
hydrophobic.
actually
Notice that's
four
phenylalanine,
proteins here
tyrosine,
that
and
makes
tryptophan.
an antibody.
Then
four
there
proteins,
are R groups that are positively
too
charged.
heavy,
There are
too
R
light
groups that are
chains.
negatively charged.
So it
So
has
right
very
away,
sophisticated
you can
structure.
tell we have 5 different
Here's
groups.
hemoglobin.
Imagine
It
the
also
combinations that
two
just
alpha and
random
two beta
proteins
subunits
can
can
have.
have.
Again, or you can have
proteins
that are
structural,
very
kind of
sophisticated structure.
A lot of these amino acids,
Things like insulin and other proteins might
a lot
have
of proteins
to be
that have a negative
altered and
charge.
changed after they're
A lot of
made.
proteins
that have a positive
and
charge.
of course it has to get to the right location
So if you think about
in the cell. So there's a lot of things that are going
DNA,
on here.
And I said there were special proteins
shape
called
and the
histones.
structure and function, but
just ask yourself, do you remember what
DNA
the nature of
molecule, and here's
histones
the
are?
lipid bilayer,
the phosphate backbone
hydrophobic
of
and the
DNA
hydrophilic
has a positive
head
charge.
groups and the hydrophobic fatty acid tails.
If you guess that
So if some of
histones
this is
tend
making
to
sense,
have a lot of
suggest you go back to
charged
your notes from
amino acids,
microbiology, basic
be,
biology, and kind of
correct
catch up
here.
Again, this
Histones,
is just
I
meant
can't
as an
spell that,
introduction.
but there it is.
Cell biology is a senior level course,
again,
some of this,
the nature
I
of
hope
the
a lot of
protein
it
has
is
to
just review
can
and
fall
you're probably
back
bored.
to
But anyway,
the
I still
structure
have
of
to
it,
try to
the
catch
so-called
everybody on the
primary
same level
structure.
before we.
So bioinformatics.
So here are
looks
building
across
blocks of
the
life.
landscape of
Different
proteins and
have
makes
different
note
shapes. There's
of
an association
positive,
with
negative
structure and
charged,
function
hydrophobic,
and
hydrophilic.
the shape of the protein.
These may indicate beginning
and
and
these are
ends
just
of
some
various
examples of
functional
different
domains.
proteins. Some proteins
And
are
so that allows
enzymes.
us to
They
make
usually
predictions
have
with
ASE
new proteins as
at
to
the end.
what is this.
Okay, here's
Where
another
does
ACE.
it hang
So
around in the
these,
cell, right?
Glutamine
So those are some
synthase
key
and
questions.
adenyl
Localize the
kinase
protein,
are
and you know
enzymes.
probably a lot
They're
about
its
also
function.
structures. These aminoglobulins
Here are aromatic
are known
amino
as
acids
antibodies,
here,
just showing.
and I know
some,
in micro
physical
you went
biochemical
into,
properties. These
detail
R
about
groups
antibodies.
tend to
It's
have
actually
an absorbance spectra here.
4
Here's
proteins here that
tyrosine,
makes
here's tryptophan.
an antibody. I'm just
4
showing you
proteins.
this again. You should have probably already
too
had
heavy,
this,
too
certainly
light
in
chains.
biochemistry. So
we'll
it has very
sophisticated
here, so.
structure.
And then here are some.
There
Hemoglobin,
are some
it also
special
is made of
amino acids,
2 alpha and 2
cysteine,
beta
glycine, and
subunits.
proline.
So these again are
Proline is known as the
structural,
kind of
chain
sophisticated
breaker.
structure.
It actually,
Things like
insulin and other
notice
proteins
this
might have
structure
to be
It actually
altered and
puts
changed after they're
dramatic
made.
kinks and twists into
And
protein
of course, it
structure.
has to get to the right location
Cysteines
in the cell. So
tend
there's a lot of
to,
things that are going on here.
form double
With
bonds with
the
each
shape
other.
and the
So you
structure
got,
and function.
cysteines
But there's some,
tend
of course,
to
here's the
do this so-called
DNA
secondary
molecule, and
level
here's
of
the
structure
lipid bilayer, the
when there's double
hydrophobic
bonds
and the hydrophilic
between two cysteines
head
in the
groups
same
and the
protein.
hydrophobic fatty acid
So
tails.
there's critical.
So if some of this is making
proteins
sense, suggest you go back to
have critical
your notes
things
from
like
microbiology,
and
basic
cysteines
biology,
in it
and
that
kind of
give
catch
it
up.
that
Again,
final
this is just
structure
meant as an
involved in
introduction.
functioning.
Cell biology
as well.
is a senior level course, so
So
some of this,
cysteines can't cross.
hope
so
a
here
lot
it is.
of
That's
it
known
is
as a disulfide
just
bridge
review, and you're probably
between
bored.
the
But
oxidation
anyway,
and
I
reduction
still have to
state
try
of
to
a
catch
protein,
everybody on the same
depending
level
on the
before
structure that a protein
So
can
here are
assume
building
based
blocks of
on
life.
the
Different
nature
proteins
of
have
the
different
environment
shapes.
the
There's an
protein in,
association with
the
structure
cysteines
and
are,
function
together
and
or
the
not.
shape
and they're
of the
just
protein.
showing. Here's a very
And
complex
these are just some
protein that's
examples
actually
of
two
different
chains. Chain
proteins.
one,
Some proteins
chain two,
are enzymes.
and those
They
are
usually
actually
have the
connected
ASE
together
at
functionally
the end.
by
Okay,
these two.
here's another ACE.
disulfide
So these
bridges. So
glutamine
that's an
synthase
example
and
of
adenyl
further structure.
kinase
It's
are
post
enzymes.
translational
They're also
structures. These
after the two
immunoglobulins
proteins
are
are
known
made.
as
So
antibodies. Clearly, if
and I
you
know
are
in micro you went into
if you are
detail about antibodies. It's actually
studying that, if you're born with
4
a mutation
proteins here that
and
makes
your
an
gene
antibody.
doesn't
4
code
proteins,
for these, cysteines,
too heavy,
then
too
you
light
have
chains.
a mutant protein and probably
So
a
it has very
dysfunctional
sophisticated
protein,
structure.
which could lead to a disease.
Here's
Hemoglobin, it also is made of
proteins are
2
peptides.
alpha and
This
2
is
beta
just showing
subunits.
the
So
structure
these
of
again
these
are amino acids here.
the
structural
last
the
amino
kind of
acid that has the
structure.
COO.
Things like
So
insulin and other
COOH
proteins might have
is
to
said to
be
be
the
altered and
C-terminus or
changed after they're
carboxy-terminus.
made.
The amino terminus
And,
has
of course,
the
it has to
amine
get to the
group
right
of
location
the last amino acid
in
here.
the cell. So there's a
So
lot of things
this
that are
is the
going
amine
on
group.
here
This is
with
the
carboxy
shape
and
end
the
here.
structure
Here
and
it is,
function.
just-
But reminding you again, that's
there's
just
some, of course,
review.
here's the
And so there's a
DNA
molecule, and
directionality
here's
as
the
protein
lipid
is made
bilayer, the
from this
hydrophobic
direction
and
and
the
the
ribosomes
hydrophilic
this way.
head groups and the hydrophobic fatty acid tails.
So proteins have
if some of this is making sense, suggest you go back
a lot
to
of the bioinformatics
your notes from microbiology, basic biology, and
predictions
kind of catch up. Again, this is just meant as an introduction.
Structural domains.
Cell biology is a senior
there
level
are also
course,
software
some
that
of this
predicts secondary
hope
structure.
a lot of
An
it
example
is
a
review,
coil,
and
alpha
bored.
helix.
But
there's also
anyway, I still
beta
have to try to catch everybody
sheets.
on the same level.
Kind of look like pleated drapes.
So here are building blocks
structure.
Of life. Different
and there are other
have different shapes. There's an association
so
with
those are
structure and function
with a higher level of structure.
Shape of the protein. And these are just some examples of different
structures
proteins.
known as
some proteins
structure.
are enzymes.
Here is a protein that may be binding
at the end,
metal ion or other
here's another ACE. So these
structure here, and you
blooming
get a
synthase and adenal
and
kinase
so understanding the primary
there are
to
also
the tertiary
structures. These
is
immunoglobulins
part
are
of
known
bioinformatics
as antibodies.
that's going on now.
And I know
so they're looking
micro,
to make predictions about mutant proteins and disease.
Detail about
antibodies.
Primary
it's
structure
actually
causes a
misfolding,
four proteins
so
here that
get
makes
the correct tertiary
an
secondary
antibody.
structure.
Four proteins,
the peptide
too heavy,
is
too light
functional.
chains.
Okay,
so it has
things like
very
gene therapy and whatnot,
structure.
or at least diagnosing
Here's
hemoglobin.
It
also
is made
replace
two alpha and two beta subunits. So these again
some other
are,
way try to cure that disease.
Structural,
and the
kind
final
of sophisticated
level,
structure.
Not all proteins
things
have this,
like
but some
insulin
do.
and other
proteins
structure.
might have to.
And this is where you
have
multiple
chains
of
proteins. In this case, 4.
And
this is
of course, it has to get to the right location
group here
in the
there
cell.
Are two
so
there's
a lot of
that
things that are going on here with.
And again, this is
the
that
shape
higher
and
level
the
of
structure
structure.
and function.
So having
there's some, of course, here's the DNA molecule, and here's the lipid bilayer.
Anywhere along the way
and the
could cause failure.
Head groups and the
hydrophobic
fatty acid tails.
And you get no function.
So if some of this is making sense,
or
suggest
low,
you go back to
low function.
It
could be
from
lethal.
microbiology,
so a lot
basic
of
biology, and kind of catch up. Again, this is just meant as an introduction.
Spontaneous
cell biology is a senior level course, so
lethality
some of
mutations, right?
Hope a lot of it is
just can't survive
and you're
the
probably
gestation
bored.
period.
Anyway, I still have to try to catch everybody
altered
on the
structure in the
before.
DNA that codes for different.
So here are building blocks of life,
and so personalized medicine
have different
will allow
shapes.
Us to
There's an association with
allow us to look
structure
at
and
structure
function and
here.
the shape of the
So,
protein.
These R
and
groups
these are
here
just some examples
do
of
have
proteins.
interactions.
Some proteins
side
are
chains
enzymes.
They usually have the ASE.
So
there are hydrogen bonds between neutral.
Here's another ACE.
So
these
there are bonding between
blooming synthase and adenal
there are ionic
kinase or
attractions
enzymes.
But there are also structures.
One
These,
protein to another. Notice the positive and the negative
antibodies.
Here. Versus two
positives
in micro,
repulsing.
you went into
then they're hydrophobic. So
detail about
antibodies.
It's actually
bury themselves and
four proteins here that makes an antibody.
That you create
four
this
proteins,
kind of hydrophobic pocket
too heavy,
here,
too light,
which might
chains.
be very critical.
So it has very
sophisticated
substrate.
Comes
in
hemoglobin,
to be,
it also
chewed
on
by
the enzyme.
Two alpha
and there's
two
also
beta
subunits.
So these again are
two atoms together can have
structural
level
kind of
weak
sophisticated
attraction.
structure.
So
things like
cell
insulin and other
level,
proteins might have to be
so far
altered and changed after they're
biochem.
made.
But we do aware,
of course, it has to get to the right location in the cell. So there's a lot of things that are going on here.
Probably change some type
of the shape and the
structure
and
these
function.
Examples of some
interactions.
There's some,
of course, here's the
to a mutation
DNA molecule, and here's the
and this is showing
the
protein A and B,
and the hydrophilic.
These proteins can come
head groups
together, so it's kind of like handshaking,
tails,
and so they can come together.
Some of this is making
and
sense.
Notice
There's
you go back to another bonds that are forming
from microbiology,
and
basic
together.
Biology, and kind of catch
and
up.
Notice
Again,
the attraction level is very strong.
Cell biology
in
is
cell
a senior
biology,
level
understanding
course, so
some pathway
hope a lot
we look
of
at
it
protein
is
A,
just review,
and we
you're
try to
probably bored.
predict
But
who it
anyway, I
binds
still
with.
have to try to
What is
catch
protein
everybody
B?
on the same level
Okay, so
before.
bioinformatics
So here
starts
with
building
one
blocks
and
of
tries
life.
to
Different proteins
figure
have
out
different
the
shapes.
other. There's an association
Hey,
with structure
barking.
and function
Okay,
and
and
the
then
shape of the protein. And these are just some examples
you
of
can
different
also
proteins.
have lesser
Some proteins
interactions
are
here.
enzymes.
Okay.
They usually have the
And notice we have a
ASE
less stable
at the end.
complex
Okay,
here.
here,
Protein A and
here's
B.
another
So
ACE.
there's less
So these
interactions.
glutamine
So imagine
synthase
these
and
pathways in the cell
kinase
signals
are
causing
enzymes. They're also
proteins to
structures.
bind
These
together
immunoglobulins
or come known as antibodies,
or become less stable.
and I know
In micro,
there's a lot of
you went into
activities that are going on,
and
we'll go
detail
into
about
this.
antibodies.
This is not
It's
the last
actually
time you'll see the
four proteins here that
interactions.
makes
In fact, the whole
an
study
antibody
of protein
four
proteins
proteins
is known
too heavy,
as
too light
proteomics.
chains. So it